Our next speaker is Amy Gladfelter, and she is an associate professor in the biological sciences department here at Dartmouth. And recently, she was awarded a graduate faculty mentoring award this year, so congratulations. And her research combines live cell microscopy and computational approaches and genetic and biochemical analyses to determine how cells are organized in time and space. And cells being the fundamental unit of life, it makes sense that we could learn some valuable lessons from them. So thus her talk is titled Learning to Balance Life from Cells.
Well, hello. It's really nice to be here. I'm really honored to be here to talk about a little bit about life and a little bit about my work. So today I have really divided this talk into 4 different parts. And I want to share with you a little bit about my path to becoming a scientist, how I decided to take on this career. And then I want to talk to you a little bit about data from my lab. I won't talk too much about that because I realize you're a really diverse audience, but I want to give you a flavor for sort of what we do, since this is a science day. And then I want to talk a little bit briefly about really what a typical day in my life is like. What is this career like, and why is it rewarding, and how do I balance it? And at the end, I have a few messages that I hope to transmit to you, which really are lessons that I've learned along the way, on the road, and that I still think a lot about and I still struggle with, actually. But sort of my years of experience have allowed me to distill this into a few key points. So that's where we're going to go today. There's hopefully something for everybody in this. And feel free to ask questions in the middle if you want. Don't feel like you have to wait until the end to interrupt me with anything. So where did this all begin? So I grew up in rural Florida. If you look at this map, it's basically between Wet and Wild, which is right above Orlando, and Walt Disney World right there. That is where I spent my childhood. And you can see from this picture that Florida doesn't really present itself as the most intellectual environment, right? It was a really fun place to grow up. And needless to say, I was not doing science experiments in my spare time as a child. I basically spent my entire childhood outside in nature playing. I was not a super studious child, and I'm saying this because you shouldn't feel like you need to have been this sort of programmed super genius in order to become a scientist. I literally spent most of my childhood climbing around in orange trees, like running around in orange groves, looking at bugs, swimming in lakes, trying to avoid water moccasins, and occasionally you would see alligators, and basically exploring the out of doors. And that is how I'm so lucky that this was my childhood. And when I wasn't outside climbing trees, such as these beautiful oak trees that are so common in Florida, or used to be common in Florida, I spent the rest of my time dancing. And I actually was an incredibly dedicated dancer. And thanks to my parents, would often be dancing almost every day of the week. And my big goal was to really become a rockette. For really the first 10 or 15 years of my life, I was planning to go to New York and dance. That was my goal. And I'm not tall enough to be a rockette, so that made that easy in a way, right? But you know, I worked hard at dance, and so I do think I learned something from this, which was determination, dedication, and you know, really structure that it took to sort of dedicate to something. But it was to art. It was not to science. And I'd say all through this time, I really had no idea scientists existed other than in this abstract, stereotypical thing you see on TV. I then went on and had the best first job you could possibly have. I worked at Universal Studios in Florida in a Star Trek exhibit, which, you know, is sort of appropriate now. I think that I actually became a scientist. And I, in this job, made movies. I filmed tourists, And they would come in and get dressed up as Star Trek characters. And I would film them in a room and tell them what to say. And at the end, they were superimposed as if they were actually in a Star Trek episode. And they got to take, at that time, a videotape, a VHS cassette, which you probably don't know what that is, but it was like what you used to watch movies on. And they would get that, and it looked like they were actually acting in Star Trek, right? And it was, I met the funniest people, right? And for this whole time period, so this was in high school and in college a little bit, but mostly in high school, I had plastic Spock ears with me at all times because I always had to put them on the different tourists. So in my purse, I always had these Spock ears. If anybody's, yeah, you probably have seen, everybody knows what that is, right? So anyway, it's interesting, this is where my movie making began. I now make a lot of movies of cells and appreciate very much the power of film, but it's a very different kind of movie that I make now. I also here realized that I didn't want to work at Universal Studios for the rest of my life, right? It was fun, but it was time to start applying myself. So it was really only around in high school that I really got my act together and started focusing on school and work, and decided I was going to do something with my life. So I was really fortunate that I was able to attend Princeton then for university. And this was a transformative experience in many ways. But importantly, I met this lady here shown in the top of my slide, Bonnie Bassler. Some of you may have heard her name. She's an extremely prominent microbiologist now. I was actually her very first undergraduate student. I was one of the first people in her lab. She is perhaps the youngest person, if not the youngest woman, to be in the National Academy. She has had an incredible career trying to understand how bacteria talk to one another. And as an undergraduate, I started working in her lab on these incredible luminescent bacteria, which you can see shown here in the top right are them growing on a plate, and here you can see them growing in a flask, and then down here you can see them growing in a squid, where they're used by this squid to cover its shadow when it's swimming on a moonlit night. So these are very simple bacteria, and she was interested in a very basic question, which is how do these cells make light? And the way in which they make light depends on them being in a community. They have to know that there's enough of them, and when there are enough of them, they glow, they make light. So they sense a quorum, and she termed this as quorum sensing. So these are tiny little bacteria that are able to talk to each other and say, I'm here, are you here? Great, okay, we're here. So we're gonna make light. And so they do that either in the free ocean or when they're in an organism like this. And what's really neat and important about this work is she was studying these very simple marine creatures. They don't matter to the world as far as anyone knows. She then went on to study this in pathogens, in bacteria that infect all of us and infect animals that we care about, plants that we care about, and it turns out bacteria everywhere are talking to each other and they're communicating with one another. And so it turns out it's very important for infectious disease, this work that she was doing, which is why she's now super famous. But what I learned from her were two things, and they've been really important to me. And one is that nature's simplest creatures, and sometimes the most basic problems, can end up being really important in the end. And I've always taken that with me in how I approach my research. I also learned from her that science should be fun and that you should always be doing other things because that's going to make you a fresher and more energetic scientist. So she was always dancing and she acted and she did all these other things that kept her a whole person. And I modeled that from her very much. And I also consider her my intellectual mother. So I still write her with frequency when I need advice. And that's one message I want to send out to all of you today is to really take care of your mentors, because your mentors can take care of you forever. She is still really someone who nurtures me. And so it's really important to have these people. So after working with her, I realized I think I really want to be a scientist. So the whole dance thing is done. I'm going to now get my PhD. So I headed south again for my PhD. And I did my PhD at Duke. And this was my PhD advisor. No. This is Coach K. So he's the coach of the basketball team at Duke. Duke, yeah, I just wanted to see if anybody was like really. But I had a picture of him above my laboratory bench because he is a famous basketball coach, right? And it's very much part of the culture at Duke. But his mantra was all about not giving up and determination and hard work. And so that is why I had a picture of Coach Pei above my bench at Duke, because my PhD, and as any of you, if you go on in a PhD, you realize basically it's not about being brilliant, it's basically about not giving up. And hopefully you can see this movie here. This is my first movie in this talk with cells in it. So my actual PhD advisor is not a basketball coach, but he showed up here in the upper left. So his name is Danny Lev, and he's my real intellectual father. And as a PhD student, we were in his lab. I was looking at how cells change shape, how they grow in one particular direction as opposed to another direction, essentially how they might have a compass that tells them which way to grow. And unfortunately, it's a little bit hard to see in this movie, but what you can see in these green circles, these are nuclei in individual cells that are sort of jiggling around, and then they're dividing and they're getting put into daughter cells. And essentially every single mother cell, it's so hard to see, but don't worry about it. Every single mother cell is essentially putting out a projection, and then a nucleus is going to get deposited into that projection. And we were studying the cell networks, the proteins that are required to build that projection, to build that daughter in one spot. And you might think, well, who the heck cares? These are little yeast cells. They're basically what make bread and beer, and like, why do I care, like how they put out a cell? But the molecules we were studying are the exact same molecules that cells, for example, in tumors use to leave a tumor. They are how a cell makes a projection so that it can crawl out of the tumor and metastasize through the body. It's the very same molecules. In fact, if you put the human proteins into these cells, they can do the same job. It's how much it's been conserved in evolution. So we were really studying this simple creature to understand how cells pick a position in space. And here I fell in love with cell biology and trying to study cell organization. I also did other things other than be in the lab, although I was in the lab a lot. I don't have a picture because I can't find any like digital pictures from this time, but I got married as a graduate student. I learned how to garden, and I took care of a very large cat who was the best cat ever. And they were all part of my life in Durham. And it was really there that I really practiced trying to have a whole life, meaning a balanced life. And we use this word balance all of the time. And I think to me it means a life where you can feel fulfilled in all the dimensions that you need, right? It's different for different people, but all the things that you need get fulfilled. And I practiced that early. I didn't say, oh, I'm a graduate student, I need to put off doing these other things. I did it then, right? And it might have been a little crazy. There were days and Saturdays where I'd run in the lab and start a gel and run back and garden, and then I'd run back to lab and do something, and then I'd run out to the farmer's market and go back to lab. It was a lot of running around, so there was a high chaos factor. But I was able to really practice being balanced, okay? And I think that was really important, and I encourage you, even starting now. And one framework you can have for this is make sure every day, maybe it's only 10 minutes or 20 minutes, but do something every day that renews you, just in some way, shape, or form, right? And that might just be a quick walk. You know, it might be a cup of tea, but like do something that renews you and just practice it so that life, it's just part of life, right? And you can still achieve, but it's sane. So I then, the next step when you're a laboratory scientist, or really almost any scientist, is to go on and do what's called a postdoc. So after you get your PhD, you go on to another lab and try to prove yourself as a scientist and develop a brand new project. And I did something that was considered and still is considered risky. And that is I went to Europe, and I spent 4 years in a lab in Europe, in Basel, Switzerland. So that's being shown right up top there. It's a city on the Rhine, in the northwest corner of Switzerland. You might wonder, how did I coordinate this with my husband? It turned out just serendipity. We both were just offered positions in Switzerland. So that's when we were like, okay, we have to do this. We were both at meetings, and people approached us and said, do you want to do a postdoc? And we thought, golly, and they were both named Peter. It was really perfect, right? Like, okay, we have to do this. Yeah.
Can you talk about why it was risky?
Yeah, I will. So right now, the academic job market is really tight. So when you, I'll go on and talk about getting an academic job. There are many more applicants than positions right now. And so it's considered important that you do everything possible to optimize getting a job somewhere, getting a postdoc somewhere where you are going to have all the connections you need and be in a super duper famous lab where you're most well poised for a job, well situated for a job. And you know, Europe was considered further away from Boston or San Francisco, sort of the two centers of biomedical research. Historically, everybody went abroad for a postdoc. So in the 70s, 60s, 70s, everybody went abroad. It was standard to go to Europe for a postdoc. And then that completely changed. And so my PhD advisor said, don't go, don't go, don't go. So I had to really go within my own intuition and say, I want to do this. This is an adventure. And if it doesn't work out, well then I'll do another one, you know? So what? I'm a little older, but I do another one. But in the end, it worked out. So it was a risk worth taking. And I'd say if for those of you planning ahead, it's a much smaller world now than it even was when I did this. The internet and air travel, everything is so easy. It's just not worth thinking twice if you can find a good research environment to go abroad. So we had a wonderful time traveling in Europe and doing science in Europe. This is a picture on the top left of who I worked for. He was my mentor, Peter Phillipson, he's now retired. And I went there and started studying these really large cells, which you can see growing beside budding yeast here. This was what I did my PhD on. And this is the creature I started working on in my postdoc. And you can see who wins, right? And I started asking these questions. How do really large cells spatially organize? So how does this cell know where it is and what it's going to do? How does it decide to put out branches at these particular spots? And it's really a question of how they organize in space and how they position things within a very large cell. And I'll talk more about that in a few minutes. What Peter did for me as a mentor was really, he gave me total space. He said, here is your lab bench, here are your cells, do whatever you want to do. And that was completely scary, but completely wonderful because I was able to prove to myself that I could do it. And before that, I sound all like I know what I'm doing. I didn't. I was really nervous. I didn't know who I was as a scientist. Like, could I really design a good experiment? Could I direct people doing science? I was really insecure about that. And then in this time, because he gave me this freedom, and I realized I could. And so sometimes you need a mentor who's not necessarily really hands-on. Sometimes you need a mentor who believes in you and is going to just let you go and really give you space. And so that's what he did for me. He's also still very close to me. I'm still really close. I didn't say this, but my PhD advisor, I talk to him every month, probably on the phone, and I'm either sharing my joys or I'm crying. So I can call him and cry because he's my intellectual father. So after 3 years as a postdoc, I decided it was time to apply for faculty positions. I felt ready to go onto the job market. That took some time. When you apply for faculty jobs, you apply to many places, like 50, 60 different places. It's a very big undertaking. And I should also say, before this, while I was a postdoc, we thought, oh, we should start our family now while we're postdocs because this is a great time to have babies because we're a little more flexible and that kind of thing. And my hard lesson I learned there was, you know, babies, you don't just go like this and you get them. You don't know when you're going to get pregnant. And so we actually tried for many years to get pregnant and couldn't get pregnant. And gave up. We just gave up trying and said, oh, we'll adopt children after tenure is what we decided. So we said that, right? And then I applied to all these faculty jobs. And then literally the week I started interviewing, I'm like, oh, I'm so tired, you know, it must be the stress. I was pregnant. So I ended up applying, interviewing for all these faculty jobs in my first trimester. So that was fortunate because I wasn't showing. I think it would be complicated to do otherwise. And so here is my son who's now 8. He was born in September 2005, and then in December 2005, my lab was birthed. It was a busy year. It was a really busy year. And so I transitioned to becoming a mother, a scientist, and a professor, like very quickly. There was just this sort of instant, like suddenly everything had changed, period. I had no time to ask questions about whether it's possible and whether you can do it all. It was happening, basically. And I can talk about this more in the questions, and I'll just talk a little bit about it now, because I think there is so much in the media right now about whether you can do it all, and I think there's a lot of different opinions out there about whether you can do it all. And I really do think you can. I think you have to give your best as you can to each faction or every faction that needs you, and have reasonable expectations that, you know, you're going to do a solid job at everything, and maybe there's a season where you're not the best at something in your career. And, you know, maybe you aren't like hand knitting sweaters for your children, but that's probably okay, because kids, nobody cares really about that when you're, you know, a grown up, that your mom knit things for you, right, or whatever it is, right? But I'm always there for my kids. I go to whatever they have at school. I'm able to do that kind of thing. And so we can talk more about this, about sort of how to do this. But for me, it was helpful that I didn't have a choice. All of a sudden, I was like, oh my gosh, I'm going to be a faculty member, and I'm having a baby. And it's all just happened. And so in a way, it was easy for me, because it was just all in motion. But it is a lot of work, but it is very doable to do it. I have an incredibly supportive husband also, and we split everything 50-50. So here I am now here. This is where I decided to take a faculty position at Dartmouth. I'm part of the MCB graduate program, and I think everybody knows the Life Sciences Center, where the biology faculty are. And so I'm going to spend a couple of minutes, not too much time talking science, because I realize we're a really diverse audience, but I wanted to share with you what I've been thinking about at Dartmouth as a scientist. And maybe part of this, even if you don't get the science and you start to glaze over at the science, maybe what you can appreciate is how much I love this stuff, right? And that is one of the reasons that I can balance, is because I have found something I love to think about, and I truly find it mesmerizing to think about what goes on inside of a cell. I think it's so totally fascinating, and I love the questions that I find in that place. And that is one key to finding balance, is finding your passion. And we'll talk about that in a little bit. So my research program has a pretty simple equation. It's basically, we take cells, we look at them under microscopes. Sometimes we grind them up and look at their parts under microscopes, but a lot of times they're intact cells. And we need people to do that. So people are looking at them and analyzing data with the help of computers. And basically, if you add all this up together, what we're doing is quantitative cell biology. And we're using very quantitative tools to try to understand how cells are organized and how they function. And so one way to think about this in terms of what I'm actually showing you here is on this side, on the left side here, is an actual cell that's growing under the microscope. It's that winner in the time-lapse movie I showed you. It's all branchy, growing all over. And you can see each of these green dots is a copy of its genome. It's its nucleus moving all around. And it's one big cell with lots of these nuclei in them. And what we did actually is convert information in this movie into a movie that here was generated actually by MATLAB, in which we have converted the cell outlines. We've had a computer detect the cell outlines. We've had a computer detect the center of all of those nuclei and chart their position all through time. And you can see, although the color is not as beautiful as it should be, the colors represent lineages of nuclei. And so they divide, and there's sort of all different lineages within there. And that information is also encoded in this MATLAB-generated movie, but it's all come originally from this movie. And we've analyzed, using various computational approaches, lots of different aspects of what's going on in this cell. And so I want to talk a little bit about sort of the biology I'm interested in, so you learn a little biology today too. And I want you to appreciate, if you take anything from this other than career stuff, that cells are extremely crowded places. They are very, very crowded places. This is sort of how everybody thinks of a cell. When you close your eyes and you think of a cell. You probably don't do that very often, but if you do, you think of this sort of textbook view of a cell, right? But the reality of a cell is, and I just need to pop out of here, here we go, is more like this. Oh. It's going to take a while. It's going to stream it. This, if you are a molecule inside the cytoplasm, it's much more an environment like this. Basically, it's like being in this, I cannot even imagine being here, but it's like, this is, I think they shut this down, but it was like a wave pool in Tokyo, I think, like in a mall or something. And it is an incredibly dynamic environment. It's probably more dynamic than this, because there's probably a lot more Brownian motion that's actually jiggling everything around and causing molecules to change places. But if you imagine each of these people as a macromolecule or a protein or lipids, membranes, those sorts of things, everything is incredibly crowded. And you're probably getting nauseous, so I'm going to return back to, here we go, this presentation here. And so here's a more static representation of this sort of tight packing that we think is actually the landscape of the cell. It's not this sort of, where cytoplasm is just this beige water. It's not just this sort of amorphous and sort of no structure to it. It's actually incredibly crowded. And the estimates for, say, a bacterial cell is that there are about 2.5 times 10 to the 6 proteins in 1 micron cubed volume. This is an incredible density here, right? So you have to imagine that life is happening in an incredibly crowded and chaotic environment. And yet, amazing things happen in this crowded environment. Just try to play this. So for example, particular proteins in this movie, one protein is labeled in red and another protein is labeled in green. And this is in the very first stages of development. And in 10 minutes, what you can see is half the cell is red. All the red proteins know to go over here, and all the green proteins know to go over here, okay? So they start out all mixed up, and it's totally crowded. And within 10 minutes, they find a place, they find order, and they find a position. They somehow find a way to go. And there's organization out of this total chaos, right? So this is a neat example. This was actually filmed here at Dartmouth by Erin Griffin, one of my colleagues. And this is the very first stages of life for a worm. And the separation of these proteins to 2 different sides of the cell is going to determine the shape of that worm. Okay? It begins at a single cell level. And it begins from that dense chaos. There starts to be places for proteins to go. So that's one amazing thing to think about, that we think a lot about in the packing and the crazy concentration of proteins. There are positions that get found. Other cells, and this is a cell like what I study, have other challenges to cytoplasm getting organized. And again, cytoplasm is this just area between all the other organelles. This is a cell that is incredibly large. You can see a scale bar here. We're cruising along. This is all one continuous cell. There are amazing currents and fluid flows going on in this cell, and yet it has to keep in charge. It has to keep order. It has to stay organized. But yet, there is very complicated geometry and currents in that cell. And here's another neat example of how cytoplasm, it's crowded, it's dynamic. It also can essentially have to have territories or regions, just like that big worm cell. Here's another kind of cell. It's a muscle cell. And each of these little tubes here is like a baby muscle cell. And muscle cells have lots of copies of the genome in it. And this is an example of how, even though this is one big cell, these nuclei are actually acting differently. They have these little spots you can see around them. Those little spots are important ultimately for that muscle to function, but you can see they're not uniform. Some regions have them, some regions don't. It's because the cytoplasm has order that's that way. And so questions we're thinking a lot about right now is trying to understand how cytoplasm is organized despite the fact that the cells are really big and the molecules are really small. It's a neat problem. There's very complex shape and fluid flows. These have to be very precise. The cytoplasm has a lot of precision about how it's patterned, and it needs to be flexible, meaning these little compartments that might get built have to be disassembled in addition to being assembled. So this is a really hard problem, and it's been neglected for a really long time because of those textbook pictures where everything's just floating in ether. Actually, there's a lot of order to that. And one of the neat models that we're thinking a lot about how this works right now is the idea that something that's very analogous to something that we all can really appreciate is happening in cells, and that is phase transitions. And so phase transitions are simply, everybody's learned this and knows this idea that molecules can go between a gas stage, a liquid stage, or a solid stage, like water, right? And that's dependent on temperature. And so there's a small number of us in the cell biology field right now testing a hypothesis that phase transitions happen in cytoplasm, that molecules go from soluble, much more crowded than this, but just sort of floating around, to a collective here, like a droplet, like water, essentially, forming an organized unit here that's going to do something. And that those same molecules may then go on and form something that's much more rigid, like an ice cube, something that's like a rigid lattice, and possibly not very good for the cell, or an aggregate. And so we've been spending a lot of time trying to test this hypothesis that maybe you get organization out of these sorts of structures assembling and moving through these different transitions here. So how have we been studying this? We've been studying this within a model system here, and this is this cell I've been showing you a little bit here and there. It's the filamentous fungus, and this is another movie where in green are the nuclei, but now we have blue flashes occurring in here. So this is a lot like that muscle cell. These nuclei, I call this nuclear anarchy, these nuclei are all in one cell, but they divide at different times. And so they create little rooms. They have cytoplasm that's their own, and it gives them directions to divide at different times. It's all one continuous cell, but yet there's organization and subdomains within it. And we've been using this, and this is, I go back to the lesson I learned from Bonnie. We've been using this as a very simple model for something I think is really fundamental to all cells, which is they have to position things in their cytoplasm and find order. And we're using this as a system to study that process. It's a model for that process. And so a little bit of data, not much, because I feel like everybody's probably a little tired. But I want to talk a little bit about work for a second. Corey Anderson is a graduate student in my group, and she has done, she graduated already and did a huge amount of work trying to understand patterns in nuclear division. And she's the one who really worked on code in order to create these sort of computational movies from the real movies. And basically all of her work led to a model where she was able to show that the motion of the nuclei were really important for organizing that local cytoplasm. So that's one key thing, is that the nuclei were putting out elbows made out of cytoskeleton that pushed apart their neighbors and basically said, this is my territory, get away. This is my territory, and they just pushed each other away. She also was able to come up with some really interesting data that had to do with, let me use my mouse here, with family lineages. And so here you can see how we're representing a nucleus that divides. And remember, this is occurring asynchronously, so that this nucleus divides, but its neighbors are not dividing. But what we did is we looked at sisters born at the same time, and we looked at how long it takes them to go on and divide, and looked at the differences in that time. And we were curious to see if sisters, they're in the same cell, do they have common timing? Do they divide around the same time? And so what's being shown here are those times, basically, where each bar has one sister in gray and another sister in black. And so there's 2 times per each line. And doing a little bit of very simple modeling, we were able to see that, in fact, sisters are more similar in their timing than what you would expect by random chance. And we could say that they seem to almost know who they are in terms of their timing. And that's related, it's inherited. And in fact, they can move very far from one another in one cell and still keep that time. So they can move to California and New York, and they still sort of have some memory of their timing. And we were really surprised by that and excited by that. And really the work of another PhD student in my lab, Chungwon Lee, followed up on that observation. And I have this picture of Chungwon. He's also graduated and moved on to a postdoc. He birthed a baby. Well, he didn't birth it, sorry. There's modern technology and there's modern technology. His wife birthed a baby, but he fathered a baby, and he is a very active father while he was a PhD student. So I wanted to show an example of balance here. And he really set out to ask the question, what are these nuclei carrying with them? How do they know their timing when they move around through this space? And could they be carrying something in their little cytoplasm, their territory, maybe that they stake out, they claim this territory? Could they then be carrying something with them as they move? And so he set out to ask the question whether parts of the cell, which are called RNAs or messenger RNAs, this is what goes between the DNA and the protein that encodes everything that a cell needs. He asked whether these might be positioned in some way, rather than just being random everywhere, that maybe the cell is putting them to create order, putting them in a certain place. And so he developed a technique where he could actually localize single molecules. So we're looking at tiny, tiny, tiny components of life here in this picture, which I think is so cool, right? And essentially each of these orange spots is a single mRNA, a single message. So we can see single molecules now under the microscope. And each blue spot is a nucleus that's making those mRNAs. And he measured where these are in cells and how far apart they are from one another. And he found one of them that behaved very strangely. It showed this bimodal distribution in terms of where neighboring transcripts were. So if he went from one and looked for the nearest neighbor, he would see this bimodal distribution instead of a nice normal distribution, which is what you'd expect if it's just random. So this was really curious. And so we ended up needing to develop some more sophisticated statistics to analyze this. So we applied a spatial statistics test to try to analyze this pattern, essentially, because we thought we had a pattern at work. And I won't go into all the details of this, but basically it was a way to look at the data, to analyze the data, and assess the position of all of the, lost my mouse, anybody? There it is. Assess all of the positions of these transcripts relative to each other compared to what it would be if they were totally spatially random. And so basically we did a lot of analyses and analyzed a lot of single cells, and using this approach we could detect whether an RNA was in a very clustered state or whether it was dispersed or whether it didn't really care. It was just all over the place. And as an aside, as a grad student, I had some famous last words about statistics. I told my husband, who is a Thayer professor, and he does statistical modeling, so he's always interested in statistics. But I was like, a well-designed experiment shouldn't need statistics, because that's what we thought in the 90s doing experimental biology, that you didn't need statistics, you did controls, right? And now all I do is statistics. It's like I live and breathe it. And that would have been very scary if I had known that in the 90s, that this was going to be my reality. But this is another lesson that, you have to embrace what you don't know, and you really just have to go for it and not be afraid, okay? So I wasn't afraid, and we've just gone for it. So basically, we were able to see that there was a pattern, which was really exciting. And we're highly clustered, a very particular thing that has this name, which you don't need to worry about. And so we were really excited, and it made it so that they stuck next to the nuclei. So we speculated that this is what nuclei were carrying with them. And we wondered, well, how does this work? And he went on to see something really exciting, which is that those RNAs bind to a protein that looks prion-like. So you may have heard of prions in the media. These are the things that cause mad cow disease. They essentially cause neurodegeneration, as do protein aggregates in general. So protein aggregates, things that stick together, are implicated in Alzheimer's disease, Huntington's disease, Parkinson's disease, prions, ataxia, and aging. All of us, as we get older, are getting clumps or aggregates of proteins in our brain. And it happens because of stretches of proteins that are highly repetitive like this, and it makes proteins stick together. But you might think, well, why does this have this? Why does this protein even have this stretch in it, right? It should be very dangerous. Turns out it's using it to actually position these RNAs. And the protein is really clumpy in how it localizes. We're looking at a fluorescent version of the protein here, and it's really like chunky. It's basically making little aggregates in the cell. But the cell likes this. It's not saying this is bad. It's not saying this is toxic. It's saying I'm using this to put those RNAs in the right location. And in fact, it matters for keeping those nuclei independent. And so if we look, for example, down here, this is a cell lacking that protein. What we can see is we're going to see all of these in purple are going to go all at once at the same time. And so this cell is using this chunky aggregating protein to create organization. Okay, so it's not just for bad, it's actually using it for good. And so what we've discovered in thinking about this is that this protein that makes these chunks, these aggregates, like a prion, is essentially using this to spatially pattern the cytosol. And this was a really exciting result for us. And we just published this late last year. And we think that this spatial patterning is really important for timing variation. And furthermore, we think it's going on and actually important probably for many different behaviors in cells that make cells different, even when they share the same DNA. And so this is something we've gone on, and we've actually seen lots of different cell processes, not just nuclear division, but also cell growth and creating new sites of growth are dependent on these kinds of complexes. And so we're really excited now in trying to understand whether this is an example of phase transitions, that these essentially RNAs and proteins that are really sticky coming together represent a change in phase. And that's what we're really actively testing right now in the lab. We're trying to understand how they're built, how they're disassembled, and actually a student in the lab is starting to see whether this might work in muscle cells, where we also see this sort of similar kind of behavior. And I don't have time to talk about any of this work, but this is just here because I have a little message that I want to give. A whole nother part of my lab is using a variety of different microscopes that have polarizers in them that take advantage of using polarized light. And with this approach, we're really trying to understand how cellular polymers are assembled. It's a totally different question. It's not necessarily related to cytoplasm organization. And this is one of the pleasures of running a lab, is you actually get to choose what you work on, and I'm working on lots of different problems. But I have this here because this is an example of polarization data here on the left, where the different colors are showing molecules changing orientation through time. And on the right right, we're actually looking at the single molecule level of polymers that we've taken out of cells and are making them like filaments totally independent of a cell, outside of the cell, in a test tube or on the microscope slide. And I have these two examples here because these are two things that I never had done before very recently. I had no idea polarization microscopy, but I sort of stumbled into it and realized it was a really good approach. And I was brave and I jumped into it, and now I'm really, really interested in using this approach. And the same with the single molecule studies on the right. It's not something we've ever done before. I've never been comfortable taking proteins out of cells to look at them. But now it's really a core part of the research that we do. And I think that many times as a scientist, you're presented with questions that force you out of your comfort zone. And that has to be one of the greatest pleasures and the greatest challenges of being a scientist, is you constantly have to keep moving. And you keep getting pushed, and you have to ride that and let yourself go to those uncomfortable places because they make you better and they make you do cool science. And so, I think the key message, one thing I've really learned, is you need to realize what you do know and what you don't know. And what you don't know should not lead you to stop. Instead, it makes you find friends. And so I have lots and lots of friends, they're not even all here, that are my collaborators that help us with the quantitative stuff that I may have never done and it wasn't my training. But I still do it and I work with them to do it. And so this has been a marvelous part of this career, has actually been working with people of all different sorts of backgrounds. And so I'll sort of spend the last couple of minutes here sharing a little bit about reiterating what is like wonderful about this career, and then some more general messages. But really, there's never a dull moment. I think those of you who know me, and some of you who do know me in the room, know that I am a very busy person. But I must say, I think I really love everything I do, and there's a real diversity to it. So on any given day, I will spend time writing. I will spend a lot of time talking to people. I'll spend a lot of time reading published papers and doing peer review, where I read other people's papers and grants before they actually are accepted for publication. In an ideal world, I would do experiments. I do very few experiments anymore, which is sad, but that's the reality of the thing that I don't have as much time for. I also spend certain seasons of the year will be heavily invested in teaching, and every day I do a lot of problem solving. And that may be the scope is broken, the computer is broken, or this reaction is not working. Why is that? There's a constant barrage of new problems. And so there really is never a dull moment. And there's a really nice mix of quiet time, where I am usually by myself thinking and reading, and interactive time. It's a highly interactive job, and that balance is really, I think, special, that I have time for both myself alone in my thoughts and then also teaching people and talking to people. And one thing I think I didn't realize is that running a lab is basically like running a small business. And now I really appreciate that. But we, as scientists, basically have to recruit people and make them work as a team. As you would in business, you have to raise money, which in the form of writing grants. You get very little money from your institution. You basically get funded through government and private grants. And you make a product, which is publishing your discoveries. And so this is incredibly satisfying. It also takes a lot of different skills at any given day. So there's also this whole management component to this job. And so I think it's important to remember when you're there at the bench, you're doing your senior thesis, you're doing your PhD thesis, and you're like transferring fluid from one little tube to another little tube, and you're like, what am I doing with my life? I can't believe this. It's like, I can't even see anything in these tubes. You know, that ultimately it's a season that you're doing that, probably, right? That, you know, if you keep at it and you're still thinking and you have good ideas, you know, ultimately you get to a much bigger picture level of thinking. You know, I'm right now thinking of 8 or 10 different projects at any given time, and they have this wonderful chance to actually be quite creative and think quite broadly and quite big. But there is a period of sort of hazing that lasts a long time, where you're in the minutia of very detailed, meticulous lab work, or, you know, It could be in computer science, you're debugging code, or there's stuff that's really tedious. But ultimately, one foot in front of the other, you actually end up opening up into a place where you do get to think quite broadly and creatively. And so, to me, these are sort of the things that I feel like I've really learned along the way as a cell biologist, and maybe some of the lessons from cells. I don't know if cells are creative. They are flexible, they're independent, yet social. They're bold and resourceful. And I think all of these are things that I have really learned to be as a scientist and as a person along the way here. So what are some of the truisms that I want to leave you with? I think my first big point I already brought up, and I want to reiterate it, and that is listen to your passion. So in this path, and you want to find a career, and I thought of this phrase, that tickles your mind. You want to find something that has problems you really enjoy thinking about, that you can imagine you're going to keep thinking about those for years and years, and they're going to change. I'll say the problems I'm studying now, I had no idea I was going to study 5 years ago, or the approaches, I had no idea, I couldn't have predicted it. But the general framework of thinking about cells and how they work, like, I love that, right? And so find whatever that is, because your mind should be happy, and it should like dwelling where it's dwelling. And I think you should also find something that's intuitive to you, because you want to find a learning that doesn't feel like work, even though it really work, shouldn't feel like work, and uses your talents, right? But it's going to be difficult, and don't view difficulty as a sign. Oh, I'm just not cut out for this, right? You hear that all the time. I'm not cut out for this. And I don't believe that for anybody, honestly. With enough hard work and enough tenacity and pleasure, if you enjoy it, you can do it, right? You may not be in the top 10%, and that is okay, It really is. There's a lot of room below 10%. The people still exist and do good things, right? So don't drop if you think, oh, I'm not the very, 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 very best, right? There's still a lot of room. And I think if you find this, if you find your passion, if you find something you really love, you're not going to want to quit or give it up when life becomes more complicated, because it will. It will become more complicated no matter what. You feel so stressed now, but it will be worse, right? And if you find something you love to do, you're not going to give it up because it feeds you, right? And so that is your challenge right now, actually, is not like learning strategies for balancing necessarily. It's actually for pure finding what is your passion. What do you love to think about? And if you find that, you can ride it for a really long time. It will sustain you, and it'll inspire you to have it all, to juggle and to run around. It may be crazy, but it'll inspire you to keep at it. And so I think that's a really important challenge for you right now, is to find your true passion. The second thing is cultivate confidence in yourself, okay? So there's something known as the imposter syndrome. It's very real in psychological literature, and it's not evenly distributed across gender. It's not to say that men don't have it, but it's well documented that women suffer from the imposter syndrome far more so than men do. And there was a great article on this recently in The Atlantic that perhaps some of you saw. If you haven't, Google confidence and The Atlantic, and you will get this. It's a fantastic article on what holds women back is sometimes just not believing in themselves. And so I can speak from complete experience here that there are many times I have been held back by not believing in myself. And not believing, making decisions because I think, oh, well, there's no way that's a good idea. I'm not going to say it. And I encourage you now to listen to that little voice, because I bet all of you have it, that says, well, that's a dumb idea. I'm not, squelch that voice, practice squelching that voice. And this quote, confidence is turning thought into action. This is from this article. I think somebody else said it, but it was in that article that I got it. That is such an easy statement, and it comes out in lots of things we do. So learn to realize when your thoughts are ending in inaction. You have a good idea and it goes nowhere. So don't let that happen. That's wasting your thoughts. So I would encourage you to force yourself to hear your voice in large groups. Ask the first question after a seminar. Ask questions in class. Hear your voice in that group to practice overcoming that little voice that's like, oh, this is stupid, I must not have heard, or I just wasn't paying attention, or I don't know, I'm not smart enough. Like, just squelch that voice and force yourself to practice squelching it by speaking up and asking questions. And don't think about what those other people are thinking, right? Think about, I am curious, my voice matters. You should listen to me, right? Who cares? How many dumb questions have you heard? Lots, right? You don't remember them though, right? You don't remember who said them. So be the person, even if it's a dumb question. Practice hearing, okay? Hearing yourself. So suggest solutions, volunteer to do things, to be leaders, and keep doing this. This needs to be a habit. And find supportive allies that nourish that sense of self. I think that's really, really important. Is to find mentors and friends that are going to help with this, okay? And that you can talk to about it. There's just a new group, this kind of applies to women in the room, but there's a lot of you, so I'll say it. There's a Lean In circle that's just started here that is meant to be a group of women talking about issues of science and issues that people face in science that have to do with confidence. And then I think this last slide sums it all up. I think you need to believe that you deserve your career. There are so many times you hear people say, oh, it's just a fluke. I'm like not really, I don't know how this happened. It's by chance. They're going to figure it out soon, right? That I'm completely not able to do this, right? So you deserve this career. It's not a fluke or luck. Don't ever say that, right? That is not something that you should ever say. It's not. You're here because you worked hard. And believe you bring a unique perspective to any situation, because you do. No one else has walked your shoes. No one else has lived your life. What you have to say is an important perspective. So you got to believe that, right? And this third point, you can do it all. I've talked about this multiple times in this. You have to have the right support. You have to have a partner, someone that will support you if you want to have kids, for example. And I think you have to have the right expectations. That, it's okay if I'm in not the top 10%, and it's okay if my kids don't have like completely homemade bread for their sandwiches for lunch, right? These are just compromises, right? There's a lot right now in the literature or in the public media, particularly from Anne-Marie Slaughter, who I think is doing fantastic work, basically sort of saying, well, maybe you can't have it all. And she's doing really important political work right now to build our infrastructure to help people have a more balanced career family life. But I would actually argue with her that with, maybe not quite enough sleep, but with a great career and a supportive partner, that you really can, and that if you put yourself in a place where you are doing what you're passionate about, you will actually be able to do it all, okay? So be true to finding something you love to do, and you'll be motivated to make it work. You won't get as much sleep as you do now, but you can sleep later, right? So it'll be okay. And this last point is don't let fear govern your career decisions, and that really wraps up all of these points, right? Believe in yourself. Don't be afraid of what could happen, right? And usually if you can cast aside the fear, great things happen. So be willing to take those risks. And so I'm just going to close with a couple pictures of my group because we talk a lot about these issues in my group. We kind of embody this. This is a picture we took that got submitted to for politicians basically to see the different faces of research. And we did a spoof picture here to represent this famous picture from a physics conference. What can you see are differences in here? What's a big difference? There's a lot more women, right? So that's great, right? We made big progress, right? So there's a lot more women here. There's lots of different skin colors here. There's lots of different types of people here. So we've come a very long way, right? It's just, we're not finished, I think, in terms of making sure everybody has equal voices. Final thing is, most important, I think, is have fun. It really should be fun. So that should be actually really a key part of your mantra is your career in science. It is a truly fun career, and keep at it. It is hard, it is difficult, it is demoralizing, but it can be very, very fun. With that, I'm going to just sort of show another picture of my current, this is almost my current research group at a party at my house, including my dog and my kids. And the people who did the work that I talked about are shown here in orange, Chongwan and Corey, and then a postdoc in the lab, Huaying. And I want to thank also the funding sources. We've been really generously funded, and that's a big part of doing science. So I'll stop talking now, but I'd love to take questions if I have time. I don't see anyone telling me, do I have time? Okay, so somebody needs to ask the first question. Hands?
How long do you, like, I don't know, at what point do you really see the turnaround where you're doing more of like that big picture work? Is it like, you know, towards the end of your, for bio, is it PhD or your postdoc?
Yeah, there's a little period in your PhD at the end where you're really reflecting and thinking about stuff. Then again, at the end of your postdoc, I would say, and then it's when you start a lab. So it's a while. It's a while. That other stuff's really important though, because it actually is like making you a careful thinker. But it's hard. Yeah, it's a long time. Yeah.
Was it difficult or kind of compromised for you to have not seen this tour?
Yeah, good question. So was it difficult for my husband and I to find the same place to work? So it was. We went on the job market at the same time, and we didn't get any overlapping offers for tenure track positions. So somebody had to compromise. And so actually my husband compromised and came here on a soft money position. And after 2 years here, he went back on the market in order to negotiate a position here. And then ultimately, a position opened up without having to really negotiate it in there in the engineering school, and he was able to get that position. But it took a lot of compromise on his part, his willingness to sort of say, okay, probably I'll get something, and if not, we would go back on the job market. So there was compromise there, for sure, and on his part. So I think those are things that's really good. I have a lot of friends that are doing this too, that you take turns, who's going to compromise, and you actually talk about that in advance, potentially. That stuff needs to get talked about in your relationship, that there's a willingness there. We were not willing to live in separate cities, so that was the baseline. So it was hard. It was easy to go to grad school. We met, I didn't say this, but we met as undergraduates, actually. We coordinated going to graduate school together, and then we coordinated going to Switzerland, and then we coordinated here. So we were a combination of luck, but then also real just decided that we were going to stay together and make it work.
And do you think that the current super competitive nature of getting funding, does that negatively affect women going in more than men?
So that's a really good question. Right now it's very, very, very tight funding in science. I mean, it's always been tight, it's especially tight now. So the question is, does it sort of adversely, more substantially affect women? I do think the competitive nature of science is one reason women leave science. And I hate to say that, but I guess I honestly believe that the culture of science is not always one where women feel like quite as comfortable. It can sometimes be extremely competitive and aggressive. And you have to really buffer yourself from that competitiveness. And unfortunately, I guess I would say I do think this is one reason we lose women. And I do worry that the culture right now is going to adversely affect women. I mean, I'm working right now just trying to talk more about tone in science so that people are more collegial in their speaking. Because right now we're in such a competitive time, it's coming out in a lot of negative language in a lot of places. And I do think that adversely affects women. So as women receiving this, you can't take stuff personally. And it's very much innately female to take things personally. We internalize criticism in a different way. Not to say men don't too, some do, but it's more common on bulk for women to be more sensitive to criticism. And that's one key thing to cultivate is just thick skin. Yeah, because the criticism will be there. Yeah.
In contrary to developing a thick skin, have you ever found situations in which you were able to take advantage of that sensitivity? Like, I mean, if it's innate.
I mean, I think I do every day, honestly.
Yeah.
Just in terms of how I relate to people or use people and try to have a sense of how they are. think it, I hope it helps as a mentor, you know. So I do think it's a total strength. I mean, I absolutely think it's a strength, and I use it all the time. Yeah.
I'm thinking about a master's in Europe, and I'm curious your experience doing your postdoc in Switzerland. What were some of the differences and maybe possibly challenges that you noticed working in European academia versus, stateside?
Yeah, I'd say European academia, there were a couple of things. I mean, it was very international, but nevertheless, it was slightly more conservative. That came out in terms of how people think or present ideas. I should say not think, but how they present ideas. People were much less willing to throw out, let's just see, maybe it's this, put something out there that might be risky or they're uncertain about. And I really discovered I like that about it. And I didn't want to stay there because it's a difficult place to go to do science. And it really still is a problem in Germany and Switzerland. And so, I mean, we had the option to stay and decided not to. So I think you have to, if you go, you just have to really be aware of that. There are different, and it's just in those Germanic places, I think that's still really a more issue. Go to France, issue, but just French.
Maybe we're out of time, unfortunately, and we have speed mentoring, and we've got faculty here for speed mentoring already, so thank you very, very